Good morning, everybody. I'm going to be speaking about sex differences in multiple sclerosis. Next slide, Carl. I have no disclosures with regards to the presentation, but I would like to note that I'm not a neurologist or an MS researcher. I'm a clinical and laboratory geneticist on faculty at Washington University. I use zebras and pictures throughout this presentation, but that is due to the way medical students are trained to consider a differential diagnosis, where the most likely diagnosis is listed first and the least likely diagnosis is last, thus the zebra in a herd of diagnoses. When I was a medical student, all of the genetic diagnoses were the zebras in the list. Lots of other geneticists use zebras to diagnose or designate genetic disorders and in their slide sets, so I'm not unique in that regard. Next slide. The objectives are to understand the current ideas about why chromosomal sex influences multiple sclerosis incidence, course, therapy, and prognosis, to learn about the genetic and cellular factors that influence MS, comprehend the types of studies that have been done to uncover why more individuals with female sex chromosomes develop MS. Next slide. So in this talk, I will be using the term sex as male or female and not gender. I'm only referring to chromosomal sex. The sex chromosomes are called X and Y, and most humans have two sex chromosomes. Typically, chromosomal sex is either XX in a female or XY in a male. Occasionally, an individual has only one X chromosome and is female, or is XXY or XYY and is male. There are other disorders or differences that can confound this a little bit so that there are in fact XX males and XY females, but this isn't a talk about that. When there's a difference in the incidence, progression, or prognosis of a disease in females versus males, that is called a sexual dimorphism. And so what I'm going to be talking about are the sexual dimorphisms in multiple sclerosis. Next. This is a normal karyotype and the kind of thing that most geneticists are used to seeing. You'll notice that there are pairs of chromosomes until you get to the last two chromosomes at the bottom. I chose to show you a male karyotype because a female would just have two X chromosomes and a male has an X and a Y, and so thus you can see the X and the Y in a male karyotype. You see that the Y chromosome is quite small and it has basically very few genes, most of which are related to sex determination. And if you do not have a Y chromosome or Y chromosome genes, you're less likely to be male. Next slide. This is a picture of zebras with their neighbors. Next. Why is studying sex differences in MS important? Well, if we look in the past, for example, in literature from 50 years ago, scientific and medical research focused on males, where the thinking was that males and females would have similar incidence, symptoms, progression, and prognosis when the subjects had the same diagnosis. Much of the time, the control group for a study were males, even for disorders that impacted women's health, except for pregnancy. When females had neurological or ill-defined symptoms, they were sometimes attributed to hypochondria or hysteria and not taken seriously. Thus, if a patient came in with tingling sensations or other symptoms that might have led to a diagnosis of MS, those symptoms were ignored. However, multiple studies have shown over many years that females are more likely to be diagnosed with and more susceptible to MS than males. Studies have also shown that males with MS are more likely to have worse disease progression than females. And so the question is, what factors influence susceptibility and progression differences in males and females? Next. So there is an animal model of MS, and it's an experimentally induced autoimmune encephalomyelitis, or EAE. In EAE, encephalomyelitis is induced in the experimental animal. Several small mammal species have demonstrated that females of the species were protected from disease while they were pregnant. This protective nature of pregnancy is also seen in humans. So does that mean that hormone levels during pregnancy are protective, or is the immune system different during pregnancy? Only research including both males and females could answer the questions raised by this observation. In EAE experiments, female animals were more susceptible to relapsing remitting disease. Thus, the animal data changed how researchers regarded female subjects compared to male subjects. It wasn't that females complained more. There had to be a biological mechanism or mechanisms that caused the differences to be observed. Next. So looking at the mouse EAE models, and I know that the figures on this slide are difficult to read, but I don't need you to read them. It's really the graph that matters. In male mice with chronic EAE, it was noticed that they had decreased numbers of neurons in cortical layer 5 of the cerebral cortex when compared to healthy male mice, female mice with chronic EAE, or female mice. So this was a concrete finding. There also were differences in mitochondrial function that were most altered in the male EAE mice. And what you see in the graph are, in the first part of the graphs, there is a very large bar showing the differentially expressed gene number in males versus females. And this has to do with gene regulation. So up means upregulated genes and down means downregulated genes. But you see in the male mice, there are many more upregulated genes than in, or downregulated than in the female mice. And so that is a concrete data point that shows that the males and the females with chronic EAE were significantly different. In addition, you see that there is a large black bar in the cortex area for the mice, and that is the bar for mitochondrial dysfunction. And you see that that bar is large and larger than the other two bars, which also have to do with neurological dysfunction that was found in the male mice with downregulation of genetic factors compared to the female mice. And again, just as in the right, the leftmost part of the graph, there's upregulation and downregulation. In the right side, there's upregulation on top and downregulation on bottom. Next slide. MS disease complexity. So MS is an autoimmune disease of the nervous system. Thus, it involves both immune systems and nervous systems in the disease progression, susceptibility, whether there's relapses and remissions. All of that involves two separate systems, which adds to the complexity of delving in and trying to figure out why there is a difference between males and females who have MS. T lymphocytes, which are a type of white blood cell, are activated and able to migrate across the blood-brain barrier. That is not what we see in non-MS patients. Cytokines, which are just chemicals released by lymphocytes, T lymphocytes in this case, cause cascading activation of microglia, which is a cell type in the brain, and astrocyte, another cell type in the brain, and recruitment of macrophages. Macrophages are a blood cell that sort of clean up debris and damaged cells, and other lymphocytes. This process leads to demyelination and neurodegeneration if it progresses. So there are actually two major types of MS, plus the secondary progressive MS, which is when somebody who has relapsing and remitting becomes just progressive. Relapsing and remitting is the most common type, and that's the type of MS where people have periods of severe disease followed by a remission. It doesn't necessarily mean that the remission is back to the initial state that the patient was in. There could be residual neurological findings during the remission that existed after the first onset of disease. But in essence, the patient has a period of time when the disease is quiet. In the primary progressive disease, there are no remissions, so that there's only a continuous state of disease, and thus it's a progressive disorder. And so that sort of delineates the differences between the initial presentations of MS. Either there's remissions that occur or it's completely progressive. A person who has relapsing remitting MS may then develop secondary progressive MS, and that's where, again, the remissions stop and you have completely relapsed disease. And then there's some very rare autoimmune presentations and MS-like encephalitis that get lumped in some of the papers. Next slide. So in this graph, Gold et al. Showed that not only were there more females with relapsing remitting MS, the females did not tend to have as much or a larger proportion of primary progressive MS. That was actually split pretty evenly between males and females. The other two bars are for MS-like disorders, neuromyelitis optica spectrum disorders, and neuronal antibody-mediated encephalitis. And you see in those two cases, they are very much predominantly seen in females. But going back to remitting relapsing MS, that's clearly predominantly female, and that's really the big question. Why is that the case? Next slide. The female to male ratio of MS incidence varies by geographic region to some extent, but it's approximately 2 or 2.5 to 3 or 3.5 now. And the ratio has only increased over time, meaning that there are increasing numbers of females compared to males with MS. Research has also demonstrated that the number of male patients with MS has remained steady, while the number of female patients with MS has increased, which is why the ratio has gone up. Potential mechanisms for this include gene environment factors, epigenetic factors, or a decrease in the number of pregnancies across the globe. Pregnancy has been viewed as protective of MS, but if women are only having 1 or 2 or no pregnancies, there isn't that protection causing them to have less likely symptoms and less likely MS. Could there be other factors that are causing the increase in females diagnosed with MS? For example, is there something in the environment now that wasn't there before that is affecting females more than males? And then has there been an ascertainment bias? Now, I mentioned that in the past, often patients who were female who had symptoms would go to a doctor and they were thought of as having psychologically caused disorders as opposed to having a real medical presentation of MS. And so is it that MS was underdiagnosed in females in the past and that there really was the same kind of ratio? We don't know. And so there's a lot of research going on. Next slide. So this is a nice graph from a paper by Bianco et al. And these are factors that can influence multiple sclerosis and other neurodegenerative diseases. And you see there's a female side of this brain and a male side of this brain. But in essence, everything in the graph in the diagram affects whether or not there is progression, incidence, severity, biomarkers that can be found, response to treatment, and all the clinical features of neurodegenerative diseases. Genetics clearly plays a part, age plays a part, hormones play a part, but there are other items in this graph that play a part. Immune responses, lifestyle, environment, nutritional habits, physiological dimorphism, that's back to the sexual dimorphism that I mentioned, epigenetics, and the gut microbiota, which is one of the more newly investigated differences between people who have MS and don't have MS, as well as males and females with MS. Next slide. And this is another diagram of something similar, but not quite as detailed. And basically, there's a question of how much is environmental and how much is genetic in the sex bias in MS. And some of the environmental factors that have been discussed in papers are vitamin D, cigarette smoking, exposure to sunlight, EBV infection, and diet. There have been papers that showed that people who, even as teenagers or children, have higher BMI and worse diets have a higher incidence of MS going forward. Next slide. So I think this slide is getting down to real science, though, and this is a question of what happens within the T lymphocyte as it is being circulating throughout a body. And you'll see that the orange is XX, and that means it is a female cell. And the blue is XY, and that is a male cell. In the female cell, you see estrogen receptors, and those are the yellow diagrams within the cell. And in the male cell, you see an androgen receptor. And that is one of the primary differences between males and females. Females have estrogen and progesterone in their bodies, circulating most of the time until menopause, and males have testosterone. And thus, the receptors within the cells are different, and the pathways that are triggered by those receptors are also different. So if you look at the E2 is the estriol and the E3 is the estradiol, they're coming into the cell on the left, the XX cell, triggering the receptor, and then putting forth a cascade of events that happen based on estrogen receptor activity. On the other side, the blue circles are testosterone. They are coming into the cell and being received by the androgen receptor and then causing a different cascade of events. In the female, you see that there is, based on the receipt of estrogen and progesterone, you see increases in immunocompetence. There's an increased risk of autoimmunity and increased immune response in response to the estrogen coming into the cell. And in fact, on the X chromosome, there are more than 1,000 genes, and those are all in 2 copies in an XX female. If you look at the male, there's increased susceptibility to infectious disease and only 1 X chromosome. So there's the 1,000-ish genes on the 1 X chromosome plus the 100-ish genes on the Y. And that's a huge discrepancy. And also it allows for a male who has only 1 X to have a disease of that's caused by a gene in the X chromosome with only 1 variant in that X chromosome, whereas a female who carries the same variant in 1 X chromosome may be perfectly fine. And so that is built into the differences between males and females, not only whether they have estrogen or androgen receptors, but also the fact that they have competent X chromosomes 2 copies, and in males, only 1 copy of the competent X chromosome. Next slide. So this is another slide from the same paper as the last, and I thought they did a really good job in putting together a review of this topic. And what you see on the left side of the diagram is the nervous system mediated effects, and on the right side are the immune system mediated effects. So in the top, you see that there could be environmental factors, epigenetics, sex chromosome differences, and sex hormones that basically across the board can influence either side. But once you get into the CNS itself, there's a neuron, there's an oligodendrocyte, there's microglia, there's astrocytes, and all of those are the cellular portion of the central nervous system. And the XYs are more susceptible to damage than the XX. In the immune system side, the XX side is more susceptible to inflammation than the males, and then the XY. And you see the different immune cells. There are T cells in this mix, there are B cells, and lots of other cells that are available to create the increased inflammatory response. So now then, if we look at the bottom, you can see that the findings are that neurodegeneration is higher in males and females. The parent of origin effect, if a female is affected, their children are more likely to be affected than if a male is affected. There's obvious sex bias, which we've already discussed. There's sex bias in just the risk for having MS. There's pregnancy reduces MS relapses in females, and then the females have more inflammation than the males. And that's sort of putting everything together as far as the clinical evidence and potential mechanisms of sex differences in one picture. Next slide, please. So, what are researchers doing that's a little bit different now than what they had been doing in the past? And that is analyzing the transcriptome or other omics, and that may help reveal mechanisms for the dimorphism in MS, the sexual dimorphism in MS. Genetic and genomic research has evolved over the past 20 years to include more than just DNA sequencing found in the genome or with mitochondrial sequencing as well. Some researchers have analyzed the transcripts, that is the RNAs found in a population of cells or in an individual, to see what is being transcribed from the genome. This data set is different from a gene expression analysis, which usually focuses on the transcripts that are turned into proteins, and also it's different from proteomics, which involves the study of the proteins themselves that are produced by the genome, but just the proteins. Another method that is being used to look at MS right now is epigenetics, or how gene expression is controlled at the DNA level. Genes may be turned on or off through epigenetics. Other methods are to look at metabolomics, at the metabolic or endocrine products of the genome, or in fact to look at the microbiome, which is often, we have microbiomes on our skin as well, but often involved in the bacteria that are living within our intestines. Next slide. So in the Catala-Senen paper, they described a flowchart of how they were going to do a transcriptome meta-analysis. And they were using data that others had amassed. So there were existing data sets of transcriptomes, and they found that their analysis revealed sex differences in multiple sclerosis. So what they were going to do was systematically review a number of papers, acquire the data from those papers, which is usually freely available. They were going to look at both blood and brain tissues. They were going to compare females, males, and females versus males. They were going to first do an exploratory analysis to see which data sets worked best. Then they were going to look at differential gene expression analyses across all of these studies. Then they were going to look at gene expression as a meta-analysis of all of the data that they pulled from the papers that they analyzed, and then look at functional enrichment, which genes were more likely when looking just at the brain samples. Next slide. So these analyses are complex, right? We're not talking about something that you can hand off to your favorite high school student and expect them to really just, be able to produce data. It's genomics, epigenomics, transcriptomics, expression studies, proteomics, metabolomics, microbiomics, and more. And once you've pulled the data, you have to ask yourself, how trustworthy is any data set? If a meta-analysis of multiple data sets unintentionally uses flawed data, the results will be distorted, and the flaw may not be identified easily. So great care has to be used when deciding which data sets to use. And if you create a new data set for yourself, for example, in the paper by Ito et al. From 2023, how well does it mesh with prior work? Is the data reproducible by looking at other data sets? Next slide. So finding the right gene or data or study is hard. And this is one of the more readily available Where's Waldo pictures that are popular with kids. But the key thing is that if each one of these individuals represents a different study, not two studies are alike. And so you have to be able to take data from study A and data from study B and show, yes, well, there is enough alike in these studies for us to use them together in a meta-analysis. Next slide. So going back to that paper that I showed you the flow chart from, the results of that analysis were, first of all, they looked at 122 papers that were screened. And of all those papers, only 9 separate studies were selected for the meta-analysis. 5 were studies performed on peripheral blood, 4 were done with brain tissue, but that's only 4 studies. Studies were not used when there weren't good representations of both males and females, when there weren't good control sets. In other words, they did not have a unaffected control set for both males and females, when there weren't enough descriptions of how the data was obtained, etc. So there were a lot of reasons why they settled on only 9 studies. From those 9 studies, there were data from 474 samples. 189 of those samples were from females with MS, 82 were from males with MS, 109 were healthy females, and 94 were healthy males. So you had a mix of both affected and unaffected and males and females in each of those categories. Several genes showed differences in transcription between the affected females and males. And I've listed some genes that they found. It's a total of 13 genes, 1 from the blood and the others from the brain tissue. And I'm not going to get into which genes they found because they don't yet know why the differences exist. In other words, they're still delving into that. They just did the meta-analysis. But they did something that they called functional studies, which isn't really like what I think of as a functional study. But what they did showed different altered immune patterns in the males and the females. Next. So this is from a different paper. And what they did was they tried to figure out the main immune features in vivo and in vitro in males versus females and in common. And they did this not just for MS, they looked at these features for several neurodegenerative disorders. But I just pulled the section of their paper that talked about MS. And you can see that the males and the females had very different factors that were assessed through a variety of papers. So in mice, the female mice had a higher cytokine production of one type, the Th1. And in the male mice, there was higher lymphocyte infiltration in the brains of the mice. But that was a little bit different in the humans. And in humans, there were differences in expressions of immune products, in numbers of T cells or B cells, and in numbers of microglia. And it was very, very different in the males. And so there are concrete items, features, differences that now can be studied because people have taken the time to look at all these studies and produce data that can now be used for further studies. So in the females, it basically, the factors that were positive were they increased inflammation, estrogen protection, and immune response. And in the males, they increased cognitive decline and neurodegeneration. Next slide. So, possible biological factors that could be different in affecting the progression rate in males versus females. We know that neurological progression and cognitive decline are more rapid in males, so that is a given. Both MRI studies and postmortem studies show persistent inflammation and neurodegeneration in males. That's not giving you a factor of why they're different, but it's telling you a fact. Mechanisms within cells may contribute to increasing damage to axons in MS, because male T cells may increase inflammation more than female T cells in both humans and in the EAE animal model. There are sex differences to how humans react to EBV infection and B cells, and EBV infection has in some ways been tied to an increased risk of developing MS. There also is genetically a male model of chronic EBV that is associated with gene variants of an X chromosome related gene. Thus, a mother who is a carrier of one of a pathogenic variant for this disorder will be basically normal, whereas her offspring who is male, who inherits that same variant, has only one X chromosome, will in fact have a high risk of having EBV-related disease going forward, including different kinds of lymphomas. Females with MS have increased numbers of CD56 NK cells in the blood, and that's something that's been shown, but nobody knows why there's more CD56 NK cells in females only with MS. There's also a difference in microglial reactivity with age when males and females are compared. And astrocytes may be more reactive in males with MS or male animals with EAE. Next. More iron accumulates within the rim of active MS lesions than in deep gray matter in males with MS than in females. And that's something that can be found. It's a fact, but nobody knows quite why. There also is a difference in the susceptibility to demyelination and repair when comparing males and females. Neurons may be more vulnerable to circulating levels of neurotoxic molecules in males over females. There are differences in the intestinal microbiome when males and females are compared. So There was a study done using mice in which germ-free mice, that's mice who do not have a microbiome in their intestines, were inoculated with the gut microbiome from identical twin pairs, humans, who one had MS and one did not. And there was increased incidence of EAE in those germ-free mice after the microbiome was inoculated from the MS twin. And that's a really interesting thing. Microbiome research has really grown over the past 10 years, and it's an amazing set of papers. I heartily recommend people looking at those. But this is something that nobody was thinking about even 10 years ago. And there's a figure coming up, and it's very complex. So that tries to put much of this together into one view. And note that when there's the term ROS, that's reactive oxygen, and that is part of what's happening within the cell. And then there also is OL, and those are the oligodendrocytes. Next slide. So I said it was complex, and that's why I started by explaining it a little bit. In this particular figure, you'll see that anything marked in green has to do with males, and either it's up or down regulated. Anything in orange has to do with females, and that's, again, up or down regulated. Things that are in italics are the mouse model. So that's the EAE model. And what you're seeing is a diagram across a brain. So the top orange line is the arachnoid. Then in the between is the subarachnoid space. And then the next orange line is the pia mater. So we're looking at a cut of the brain. You're seeing B cells and T cells on the outside. You're seeing plasma cells on the outside clumping together and sending immune molecules across into the brain, across the blood-brain barrier. You can see in males it decreased oligoclonal bands and increased in homocysteine that's coming in through the blood vessel that's within that same space. Those little light purple cells are the astrocytes that line the inside of the brain. And then you see that you're getting iron release from cells that is then contaminating the other cellular material, including the neuron, that the axon is getting chopped up because of some of the activity of some of the immune system and enzymes that are released. The little green circle that looks like teeth in the middle of the yellow neuron, well, that's mitochondria. And in females, there may be increased mitochondrial function and increased resistance to those reactive oxide substances. And so you see that there are a whole host of things that can be going on if you put it all together in one picture looking at a model of the human brain. Next. So there are sex differences in treatment opportunities. In Austria, there's a nationwide registry of MS patients. The cohort is more geoethnically similar than in studies performed in diverse areas. It's Austria. It's one sort of geoethnic ancestry across much of the country. So definitely you're not seeing diversity. However, in a study analyzing data from 4,840 individuals with MS in their nationwide registry, more females stopped both highly effective and moderately effective therapies early by choice. For the moderately effective treatment, most of the individuals who stopped were younger, and the rationale for stopping was the desire for pregnancy or for nursing a child. They did not want to have the medication cross through to the breast milk, and they did not want to expose their fetuses to whatever the medication was. There was no frequency or difference, I should say, in the female to male de-escalation frequency. In other words, going from a highly effective to a less effective medication. Either people stopped or they didn't, or they were escalated in their therapy because their symptoms were worse. Females who stopped the high efficiency therapies more frequently also had disease progression, adverse events, or other medical issues. Next slide. We needed some more zebras to let us think for a moment. Next slide. So, words of caution. I've now read so many papers in this area, probably about 50, and I want you to know that we know MS is a complex disorder and that there are sex effects in MS that may be categorized in a number of ways. Incidence, increasing sex bias over time, rate of progression, parent of origin differences and inheritance of MS-associated genetic alleles, protective effects of pregnancy on MS, differences in immunological function in males and females. None of these categories of sex effects is straightforward, leaving many avenues of further research. And I have to tell you that the majority of papers that I pulled were review articles as opposed to the primary research because there were thousands of papers. Next. So in summary, significant sex differences are found in patients with MS. Some of these are related to young women's opportunity for having children as well as the protective nature of pregnancy. Many studies have been performed looking at all aspects of MS in relationship to sex differences. So ratio numbers of males to females, hormone levels, immune markers, rates of progression, rates of neurodegeneration, and differences in gene expression and cellular processes. Many questions remain. Next slide. Thank you. Next slide. I would like to thank Dr. Chaswell and Brain Health Alliance for inviting me to give this talk. I would also like to thank all of the MS patients, their families, and the clinicians and researchers who contributed to the studies I have referenced, and actually all of the mice, rats, and other small mammals who participated in the EAE studies. Next slide. First page of references, too small to read. Second page of references. Oops, and that's that. So 1 and 2. Thank you all. Are there questions?
Yeah, thank you so much. That was a marvelous talk, and I have to get my GoToMeeting back so I can see here. Who has questions? But let me first again thank you. I really do hope that you will put together your discussion in a review with commentary. So I think it would be marvelous to pull all that together, and then also if we could get some others to help you by bringing in mention of radio tracers in the development pipeline that might be targeting the different pathways for imaging. That would be marvelous. That would be amazing. Who has questions?
Oh, go ahead.
Go ahead. After you.
Thank you. Hi. Yeah, so great talks, very interesting. I had a question. So you mentioned that estrogen seems to be more protective. Is there any difference in the relapses in MS in menopausal women?
That's interesting, and that's something that's been studied. It's not the relapses, but the progression. They seem to follow sort of some similar similar kind of progression as some of the males do. Once the menopause occurs, there isn't estrogen around. And so, although there isn't the same testosterone level as you might find in a male, there's not that same estrogen crossing into the cells and creating the immunopathology that we might see. And so, yeah, it's absolutely known to be different.
Very interesting. And then also another follow-up question. So the multiparity in women, is that protective aspect shown to be less of an effect after the pregnancies, or is it also during pregnancy that they don't seem to have as many symptoms?
During pregnancy specifically, but after pregnancy as well for a while. And it all has to do, I think, with the immune response and hormone levels at that time. And if you think about it, women's immune system has to be able to tolerate the presence of a parasite. The fetus is a parasite and of a separate genome that may be half mother but not fully mother. And so, there has to be something that allows a woman to remain pregnant. And there are studies within the realm of infertility looking at immune responses that a mother might have to a conception that prevent pregnancy. So it's very complex.
Okay, thank you very much.
You're welcome. Adam, you had a question.
Oh, yes. So can you tell us a little bit about how you'd think of EAE model, the results from the EAE model as relating to human results? Like, what are the caveats? What approach would you take if you learn of an interesting result in the EAE model? What would you do with that?
So that is a really good question. The problem with EAE, right, it's an encephalitis that's caused in an animal that doesn't really have an MS phenotype by itself. So there's always the question of whether what you're seeing in the experimental animal is translatable to what you see in humans. And so every single study that uses data from EAE studies will have that caveat built in that, well, this is found in the animals, we don't know if it's found in the humans. And there are things that are found in the animal model that are not in humans, and there are things found in humans that are not in the animal models. And so there are papers delineating some of those differences. It's hard because if you don't have an animal model that's readily available, you're sort of stuck using the EAE animals.
Okay, thank you. Also, in general, thank you for this very wide-ranging and well-researched presentation you've given. And though I do have one other sort of more technical question. So in the slide sorry, just a moment. Yes, so in the results of deep transcriptome meta-analysis, you referred to the data being used for functional studies in quote marks. Is this one of those functional enrichment analyses where they look at the gene list and look for enrichment of terms in the gene ontology, or is it different? Because you pointed out it doesn't match up to your idea of a functional study.
Right. It's not as if they went back to the cells of the patients to see that there was a functional change. They went to the data sets and looked sort of in a way at the paths and pathways through cellular biochemistry that the different genes were in. In other words, if there was upregulation in a gene that was in an immune regulatory pathway, that was a functional change in their mind. And that's what they looked at.
Right.
So it was completely in silico as opposed to being in vitro or in vivo.
Right. And these studies are quite interesting insofar as they leverage a large amount of past research to arrive at a sort of general picture of
Mhm.
what these genes in the gene set are relevant to, but there are limitations on what you can tell from that versus from looking at the individual person.
Absolutely. And I should say, though, that there were some real leaders in the field, people who had put together numerous papers in the past 10 years that really changed the view of the whole of MS research. There's a group at UCLA that's done that. There's a group in Toronto that's done that. And several of the papers that I referenced in the talk were definitely from review papers that they put together talking about all of the research. But, for example, the paper by Ito, that's from the UCLA group.
All right. Well, thank you very much.
Yeah.
All right. Well, I want to thank you again. This was really a marvelous presentation, and I'd like to share a reminder of some of what we do at Brain Health Alliance with our holistic approach based on some of our core foundational questions about same, similar, related, and different. About our approach where we look for frameworks where we can do compare and contrast. And some of you know that I've been following these stories for a long time, my entire career. And I must say that this is really, really exciting for me. I think we are really finally going to have, I usually try to avoid hype, so please do not interpret this as hype. But for the first time in my professional medical scientific career, I believe that we will be able to pull everything together in the next 5 to 10 years, especially when we look at these kinds of contrast and compare frameworks, as we can do with this male-female um, uh, approach to looking at differences as applied to the context of the disease commonly known as multiple sclerosis. Uh, I began my career at the beginning with biochemistry and immunology. I did a good number of papers on cytolytic T lymphocytes. I really do believe that everything is going to come together in the next 5 to 10 years, combining everything that we now are pulling together from genetics, from what we're learning about the immune system, from what we're learning about the nervous system, from about what we're doing with molecular imaging. And yes, I do agree with you completely. We should continue to use the word functional when describing and documenting functional activity in a live person, a human being, and or measured biological activity from that real patient or human being. I agree that we should not use the word functional when referring to purely data meta-analyses done in silico. That to me, I agree, is confusing and misleading. But I do want to emphasize my real optimism. Um, and so this should be a very exciting next few years of clinical medical research that should give our MS community real hope that we are finally going to be able to pull things together in the next few years, certainly the next decade. That would be my expression of hope for the future. So with that, unless there are any other questions, I suggest we take a break to give everybody a chance to reset, and we can get back on a schedule with Dr. Adam Craig's talk at 10:30.